Well,
Yep.
what's up?
Not much. How are you?
Same old. I was on the phone with my mom yesterday. She always asks me, she's like, what's new? I'm like, you asked me the same question. Just studying this exam and it's
Yeah.
really it.
Yeah. The other hurricane missed you too.
Yeah, dude. It's so funny.
Yeah.
Some of my New York friends are asking, like, are you good? Is everything okay? Today is the best weather we've
Mhm.
had in the last week.
That's crazy. Yeah.
Last night it was actually very, very windy. I was getting a little scared.
Yeah.
Right now, though, t the whole day today has been amazing.
Yep.
Um, but hopefully you don't get hit. From what I hear, like north of Miami, it's bad.
Hmm.
So
Yeah.
yeah. So far, I mean, I hope I didn't jinx us, but yeah.
Yeah. All right. Yep. What would you like to work on today?
Yeah. So I went through the entire Biochem diagnostic that I took on Jack Weston, and I have a couple of problems that I wanted to go over. And also just wondering, like the recent sessions that we had, when are they going to be updated on YouTube, do you think?
Um, I mean, like I have like them uploaded like on like a schedule, so
Okay. Okay.
but you could also, yeah, record this one.
Okay.
Mhm.
See it? Right.
Mhm.
So this is the first question I got wrong.
Okay. Um, so how did you um go about this?
Yeah. So, um, when I do know what temperatures that you some enzymes work well at high temp, like I mean, at high temperatures, a lot of them get denatured. Sorry.
Mhm.
Um, yeah, I don't know. That's really it.
So, uh, so think about what does, so let's just say you have any type of, you know, let's say reaction or something like that. Um, Increasing the temperature would do what to, like, let's say the rate of a reaction? Like, not increasing it to the point where, it denatures, but
I mean, it's gonna speed it up.
yeah, good. So, so okay. So we should see an increase in Vmax in that scenario, right?
Yeah. Yeah.
Um, and then yeah, if it's too hot, it can cause the enzyme to be denatured, which would then have an opposite effect, causing a decrease in the Vmax, right?
Yeah.
Okay.
Wait.
Yeah.
Oh, okay. Got it. So that's why B might be an answer too.
Hmm.
Okay.
Yep. Um, and then for the Km, like, so okay, so I guess, you know, if the temperature is going to make the reaction proceed faster, that means that enzyme and substrate will bind to one another. The enzyme will turn the substrate into a product, and then it'll release that product. So you're going to have more enzymes separate from their substrates. And that could also mean that the concentration you need to of substrate that you need to saturate the enzyme will go up as well. So that can also result in an increase in Km.
So you're saying because it's not because like increasing the heat is going to disrupt the binding between the enzyme and the substrate.
Uh no, I mean, because that would just be something. I mean, that could be something that happens if you make it like really, really, if you make it hot enough to maybe, denature the enzyme. But well, if you denature the enzyme, it's not going to even be really able to function well. Um, but I think so. Like the Km, like that's the substrate concentration at 1/2 Vmax, and it kind of is a kind of like a measure of like binding affinity, or rather like dissociation. So yeah, so if you have just any kind of enzyme substrate reaction take place faster and faster and faster, um, you know, it's the enzyme is not going to be holding on to that substrate for too long. It's going to turn it into a product and then, let go of it. So you're going to have a lot more separation of enzyme and substrate, or substrate and enzyme and product. So yeah, it would require a higher concentration to saturate it.
Hmm. Okay. Um, so basically, just because it's working, the enzyme is working faster, you need more of the substrate.
hmm Yeah, because I think so I could think what could be confusing is like, okay, we know a competitive inhibitor increases Vmax, increases Km, and that seems like it's a bad thing. But when you don't have an inhibitor presence and you increase the Km, it just means that uh it's able to um it's more difficult to saturate it, which means that, and saturating it means, you're giving it the maximum amount of substrate that the enzyme can process, per unit time. And so if you increase that substrate concentration needed to saturate it, it means that because once it's saturated, it's maximum, you know, kind of ability for that enzyme to turn the substrate into the product. So if you increase that, it means that it's going to be less saturated, meaning it's going to hit its limit a lot less.
Okay. What's this?
But yeah, I guess a better reason would be what you were saying. Like if you increase the temperature beyond such a point at which it denatures the enzyme, it's not going to be binding very well to the substrate, and therefore will need more substrate. So that's a better explanation for why the Km increases.
Yeah, because that's what it said. I actually didn't even realize that's what it said, but now that I'm looking at the answer, it said it in the explanation too.
Okay. Yep.
So, yeah.
Got it.
Okay.
Oh yeah, so this is um a high yield topic here that deals with kinases.
Yeah.
So kinases, what can you tell me about kinases?
Kinases add phosphates
So,
through ATP. They take a phosphate from the ATP. Um, a kinase takes a phosphate from ATP and adds it onto a molecule, which leads to ADP. and the molecule being phosphorylated.
All right. Now, what enzyme class does kinase fall under?
Um, Ligase.
So the thing is that
But I forget. It's not ligase. Okay.
so so the thing is that while it's true that okay, so a kinase is a transferase, right?
Yes. Okay. Yeah.
And so now contrast that with so a phosphorylase would phosphorylate something, right? Um, and a phosphatase would dephosphorylate something, but a kinase is just a transferase, which means that it just transfers phosphates. So it doesn't have to necessarily always add a phosphate to a substrate. It could remove a phosphate from a substrate and give it back to ADP to make ATP. For instance, glucose to glucose 6 phosphate, the first step in glycolysis is the hexokinase, right?
Yeah.
And that's where you take ATP, that's where it takes a phosphate from ATP, resulting in ADP and glucose being phosphorylated here, right?
Hmm.
So some steps later, you have PEP, which is phosphoenol pyruvate, and you get pyruvate from that. And this is pyruvate kinase. Now, here we're removing a phosphate.
Yeah. So why isn't that reversible then? The pyruvate kinase step is not reversible.
Oh yeah, that's for, that's going to be for like a different reason. But what I want you to see here is just that it can also remove a phosphate from the substrate,
Yeah. Yeah.
give it to ADP to produce ATP. So it can do either or. So that's why it's a transferase, not necessarily something that only adds and subtracts or removes. Now, what also is important to know about kinases is that they need to use uh, three particular amino acids, STY.
Yeah.
Do you remember what these were?
Serine 3 in the entirety.
What do they all have in common?
OH.
What do I always say about OH groups?
It's phosphor. It can be phosphorylated.
So like yeah, but also it's the most common nucleophile you'll see on the test.
Oh, okay.
Whenever you see a hydroxyl group, think nucleophile. Most common
Okay.
nucleophile would be a carbonyl group because a carbonyl group involves a carbon double bonded to an oxygen. And since the oxygen is super electronegative, it pulls electron density away from the carbonyl carbon and towards itself. So that means the oxygen has a partial negative charge. The carbonyl carbon has a partial positive charge. And nucleophiles are just Lewis bases, right?
Yep, yep.
Which means that they are what?
Electron pair donating.
Yep. So this will attack that, momentarily push that up there. It'll come back and then it'll kick off that leaving group. So hydroxyl group, most common nucleophile, carbonyl group, most common electrophile. Now, what about a phosphate? Now, the phosphate it looks pretty similar to, you know, a carbonyl, right?
Mhm.
So it can also nucleophilically attack the phosphorus atom here, push this over there, push this back here, break this off. And that's how, serine, threonine, or tyrosine can be phosphorylated.
Wow. Okay.
Now, this is going to be, and this is just like adenine, or rather, the ribose sugar attached to the adenine. but this would be ATP, right?
Got it.
Now, going outwards from the adenine, we designate the phosphates. The closest one is alpha, followed by beta, followed by gamma.
Yep.
And so kinases, right? If they like, not when they do this, but when they do this, meaning when they take a phosphate from ATP, they
Hmm.
take the gamma phosphate.
Hmm. And that's just a known fact. You just gotta know that.
Yep. So
Got it.
everything about kinases is super high yield. So you want to know the following. Kinase is a transferase, which means that it just transfers functional groups. Doesn't necessarily add them or subtract them. It could do either. They require STY because those are the three amino acids that have hydroxyl groups as their side chain, and the hydroxyl group is a very, very good nucleophile, and a carbonyl group is a very good electrophile, and a phosphate happens to look like a carbonyl, and therefore the phosphate can be the electrophile here. And so the hydroxyl nucleophile can attack the outermost phosphate and, pulling it out and attaching it to itself, and then it can attach it to something else. And that's the gamma phosphate, yeah, the outermost one. That type of notation exists for other things too. Like if I had something like this, like let's say something like that, like this would be like the alpha, the beta, the gamma. Like as you move outwards from whatever site of importance, it'll just go in alphabetical order, like Greek alphabetical order. Yeah.
So kinase is a transferase and it requires one of the three amino acids, STY. And
Mhm.
like so when it kinase phosphorylates, it attaches the gamma phosphate from ATP to one of STY.
Yeah. And then the STY can give that phosphate to some substrate. So in this example up here, the STY that's attached to the phosphate gives it to the glucose to turn into the glucose 6 phosphate. Yeah.
Repeat? Wait, what?
So so the kinase is going to take the kinase is going to have STY take the gamma phosphate from ATP and then give it to whatever substrate it's acting upon. So in this hexokinase example, the substrate is the that it gives the phosphate to is glucose.
Okay.
Yeah. So it takes the gamma phosphate from ATP and gives it to glucose to make glucose 6 phosphate.
Okay. Wait. Don't delete this thing yet.
Mhm.
Making an Anki card.
Mm. Yep.
And the additional information I wrote. Asterisk gold from Shane. Asterisk. All right, perfect. Next.
Mhm.
Let's see.
And sometimes they might ask you about phosphomimetic,
Mm-hmm.
and that just means some type of, let's say, like some type of amino acid that can function as if it had a phosphate, aka would just be an amino acid that's negative. So like, because when you phosphorylate something, you impart a negative charge on it. So so the phosphomimetic amino acids could be like the acidic ones because those will turn negative.
Okay. So a phosphomimetic amino a phosphomimetic is just a negative. It's something that's negative. That
Yep.
acts as a negative amino acid.
Yep.
Okay, next. So this, yeah, I feel like it's pretty important to know.
Um, yeah. So, okay. So the thing with cholesterol is that it can do the opposite of what. So, like for instance, for Roman numeral two here, um, at low temperatures, the cell would want to be more fluid or more rigid.
At low temperatures,
Yeah.
uh, I don't know.
So let's say you had something and you make it, let's say that you just have like some soda or something and you put it in the freezer. Does that soda get more
It's a good
rigid?
analogy, actually. Yeah, more rigid.
Mhm. Good. So at low temperatures, things become more rigid, and cholesterol would act to reduce that rigidity and increase the fluidity. And if something's at a higher temperature, it would become more fluid, right?
Yeah, yeah,
And cholesterol in that case would work against that to help make it more rigid to counter.
right.
So cholesterol in the plasma membrane acts to kind of buffer the rigidity and fluidity that come along with like low temperatures and high temperatures. So Roman numeral two says increasing the amount of cholesterol in the plasma membrane at low temperatures, that should increase fluidity. So, and the question's asking what would cause decreased fluidity. So Roman numeral two would be the opposite. If it's at high temperatures, right, then that could be an answer choice. But at low temperatures, it'll do the opposite. So Roman numeral two is out, right? That makes sense?
Yes. At low temperatures, cholesterol
Mm-hmm.
increases fluidity.
Mm-hmm.
Yeah. Okay.
It'll just do the opposite of what would normally happen without it.
Yeah,
Okay. And then we have um increasing the number of unsaturated phospholipid tails. What do they mean by unsaturated?
yeah. Unsaturated is there's double bond, which means there's like a kink. So yeah, I mean it's just double bonded. Saturated is it's no, it's just single bonds.
Okay. Um, which so tell me how those can affect, let's say, fluidity.
Yeah, I mean I think from what I know, when you have unsaturated, so when you have that double bond, it increases fluidity.
Mhm. Yep. Why do you think?
That's the thing. I'm trying to conceptually wrap my head around it, and I don't know. I mean, what? There's more space. Like it's harder for them to move. That's the thing. How does it increase fluidity if it's harder to move? Like when if there's double bond. Conceptually, it's tough. I don't know.
So typically it could be like, let's say something like, well, something like this, if it's like a cis bond or something, like
Hmm.
it would create like a kink here. Um,
Yeah.
but even if you didn't have that, like let's say, let's just say something like this compared to, this one. Um, Without the double bond, you can pack molecules of this, single bonded, tail much more closely together compared to if you had a kink from like a double bond.
Hmm.
So it's easier to pack when you have saturated tails um, compared to unsaturated. So saturated, so if it's easier to pack together, it can be more solid, more rigid, less fluid, right? Because like if you think about it, like rigidity, like is when something can become more of like a solid. Like at low temperatures, things become so like water, right? Low temperatures, it becomes more rigid because it can form like ice, right?
Yeah.
And high temperatures, it'll be more fluid because you're gonna separate those intermolecular, break those intermolecular bonds. So think of it like this. So like rigidity, if something can be more rigid, it can be more solid. If something can be more solid, it can be packed closer together. Whereas, and the example they usually give is like butter, which is solid at room temperature, but like olive oil has unsaturated, double bonds in it, which make it so that it's harder to pack together. And if it's harder to pack together, it's harder to turn into a solid. It's harder to make it rigid. So it will end up making it more fluid.
So saturated bonds make it more solid.
Make it more uh solid, you said?
Yeah.
Hmm. Yep. So you can see kind of like, you know, oops, here in the chat, I put a picture.
Yeah.
See if you could show it. I think you would have to share your screen instead of just like the window.
Yep.
But
Um,
yeah. So so okay. Like I'm gonna use pens here. So like let's say that we have single bonds. Like both of these pens are like single bonds.
mhm.
If they're single bonds, they can rotate around like freely. Like it's kind of hard to see, but the top of my pens with the cap up here,
Yeah.
you see what I'm doing?
Like
Uh,
you show me your pen.
yes. So the top of the pen is like the cap, so you can see
Yeah.
that it can. So like if I had two single bonds here,
Okay.
they can freely. You see how it rotates?
Hmm.
Single bonds can freely rotate like that. Um, so if I had, just another single bond thing, I could they could I could just put one on top of the other and they can pack together, right?
Yeah.
But let's say now that we had a double bond. Now let's try let's see if these can freely rotate. Can't, right?
Nope.
Double bonds can't freely rotate. So they're going to create kinks. And you can see this in the picture on the left. You can see that the unsaturated hydrocarbon tails with the kinks, like are not like close, can't be closely packed together because once you introduce a double bond, it's going to go in a certain direction and can't freely rotate or go back to like the opposite direction or anything like that. So you can't pack them in as closely. So, and a solid is, going to be more packed, closely packed, and thus, more dense. And that's why, if you have unsaturated phospholipids or, unsaturated, fatty tails, like in oleic acid, like olive oil, it's gonna be more fluid. Whereas if it's saturated with hydrogens, it can be packed closely together. And that's like the example sometimes that they like to give is like butter. So that makes sense.
Yeah, I guess kind of. I just always thought of fluid being more like
Hmm.
because it's single bonded, like
Hmm.
it can move around more, which makes it more fluid. But I need to get that out of my head. That's wrong.
Um, yeah. Yeah. You want to think about it as how can I, how easy would it be to turn this into a solid, right? So
Yeah.
you have, straight lines or something like that. Um, you can just pack them in closely together. But if you have something that's just like bent in some other direction or something like that, uh, you can't really pack them in tightly together. And so that means that rather than seeing a fluid as something that can freely move around, think of a fluid as something that, because of how it's made, can't be compressed any further.
Hmm.
So think about it more so like that.
Yeah.
Yep. So increasing the number of unsaturated phospholipid tails would increase fluidity, right?
Yeah.
All right. And then Roman numeral one says increasing the length of the fatty acid tails in phospholipids, right? And, these are, if they, if they're not saying that it's unsaturated, then it's essentially meaning that it's saturated. But since the fatty acid tails are the parts that are not polar, aka hydrophobic, the longer the length of the fatty acid tail means that it's going to be less soluble. It's less water soluble.
The longer it is,
Yep.
the less fluid.
Yeah. Mhm.
Is that because of like Van der Waals, force?
Um, so the I don't know why I'm getting all these.
They're interacting with each other more. There's longer and more Van der Waals.
Sorry, I don't know what's going on here. Jesus Christ. Okay, I don't know what that was, but um, yeah, those tails are not like in a phospholipid, right? The tails are the parts that are not water soluble, right? Like in the picture, the circle thing,
Yeah.
circles are the phosphates of the phospholipid stuff, and the fatty acid tails are the ones that are, in the middle of that. bilayer, and if you increase, and they're just by nature lipophilic or hydrophobic or nonpolar, right? So because they're just hydrocarbons, which are hydrophobic. So if you just made that hydrophobic tail longer, it's gonna just be even more hydrophobic, um, which would lead to yeah, reduced fluidity.
Hmm.
Mhm.
Yeah. What are the main like in terms of like sphingolipids, phospholipids, all that, what are the main things we should know? Like I know sphingolipids have sphingosine and they're like amino based, amino,
Yeah, yeah,
what's
yeah.
amino?
So yeah, so your typical, your typical lipid would be a like a triglyceride, for instance. And,
Yeah,
um so you have glycerol, which is this, and let's say you had three, you know, fatty acids, which would just be like a carboxylic acid end, and then a long, like, a hydrocarbon tail. And then, these can come together to give you this, right?
yeah.
Like the fat would be like functional group would be an ester. So this is just a regular triglyceride, right? Now, let's say we had two of these fatty acids, but one, let's say this one, we just turn into, or we make that a phosphate, then that is going to be, your phospholipid, with the phosphate being the circle part that's, facing the outside of the cell and inside of the cell, which are water. So that's, going to be your phospholipid. Um, Now the sphingolipids, rather than using um the, what is it, the glycerol backbone, it's going to use the what was it, the amino alcohol, I believe. It's going to be, whoops. Yeah, so instead of it being based on a glycerol backbone, the backbone is going to be this amino alcohol uh functional group. So that's what you're really replacing. So like essentially, like this part, the fatty acid part, doesn't really change. Um, but this part is what's different. And that would be like something like an OH and like an NH, um, something like that. NH like two, and then maybe like another carbon with an OH, something like that. So that's going to be what's different for um like a Sphingos thing.
So like it just has a nitrogen.
Sorry?
It has the nitrogen.
Yep. The backbone is what is different. Yeah.
Okay. Um, yeah, this is next. But I think I got this right. Confused. Okay. Yeah, I got it right, but my question, I guess, was
Oh, yeah.
I mean, this is just like Le Chatelier principle.
Mm-hmm. Yeah. Um, so yeah, a little on the tough side because they're talking about something that has a positive delta G, which means that, it's not spontaneous, which means that it's not an irreversible step as we know it, right? But they're just asking if it's not an irreversible step, why does it still move forward, right?
Yeah, and like I get why it's C, right? But like it why isn't it A? Because if it's positive change gives free energy means it's not spontaneous.
Hmm.
And usually reactions that are not spontaneous, don't they use ATP?
Yeah, if it's going to, yeah. So okay, if something's not spontaneous, in order to make up for that positive delta G, it can hydrolyze ATP and therefore have that energy. Um, but it's nothing really here is going to be coupled to ATP hydrolysis. So we want to think, okay, how else can it proceed, right? Um, and that could just be through a buildup of the reactant and Le Chatelier principle.
Mhm.
So, like, yeah, so like, the stuff that would require ATP hydrolysis would be like, um, well, first of all, it would require something um, like a hydrolase or something like that. Um, so, and yeah, nothing in glycolysis involves that. Like, because you have your kinases in glycolysis that move, transfer phosphates and stuff, and you have a bunch of dehydrogenases that carry out redox reactions, but nothing, yeah, we're not doing anything with ATP hydrolysis
Mhm.
in any of that. But in the Krebs cycle though, the succinyl CoA turning into succinate.
Succinyl-CoA to succinate, succinyl-CoA synthetase,
Uh-huh.
GTP,
So
GDP to GTP, yeah.
the synthetase, right?
Yeah, yeah.
That is when you use energy from the hydrolysis of ATP, yeah.
Okay.
So yeah, that question likely was kind of like testing whether, in glycolysis if there's any, you know, ATP hydrolysis or not. So you would have to know that there isn't any. There's a bunch of kinases, there's a bunch of redox happening. Um, and then isomerases, which is just moving stuff around, but there's nothing and that involves ATP hydrolysis and glycolysis. Yeah. Good. Okay. So, oh, okay. So similar kind of thing. Um, okay. What are your thoughts here?
Yeah, I know phospholipids don't remain in relatively fixed positions. They usually move around
Hmm.
pretty often.
Yep.
B, like I don't know what leaflet even means. Is that like the fact that there's a top and bottom? Like there's two halves of the phospholipid layer.
Mhm.
So like they can kind of jump from the top to the bottom one.
So yeah, so you could have your you could have things moving around on the top layer um that stay in the top layer, and then you can have things moving around that can move from the top layer to the bottom layer or vice versa. And we can call them the two types of movements different things. What that show up in some of these choices. So what what would you think?
Yeah, I don't know, honestly. I'd never really studied that much. I mean, if it's trans bilayer moves to the other bilayer,
Mm-hmm. Yeah. Well, so what does lateral mean?
that it moves to like another spot on the same leaflet.
Yeah, right. Lateral movements like moving sideways.
Yeah.
So let's say phospholipid A here moves here. And let's say phospholipid C moves here. And that should be lateral diffusion. Um, but let's say this is phospholipid X, and let's say it goes and moves here. Now that can't be lateral, but it could be trans bilayer diffusion because you're going from one end of the bilayer to the other end. Which do you think would be easier?
I think it's easier to move laterally.
Correct.
Um, then just use the answer.
Wait.
Wait, D's answer, sorry.
Yep.
C is incorrect.
Yeah. So we know A is absolutely not true. Doesn't stay, remain in fixed positions. Definitely not true. Phospholipids remain in only one of the membrane leaflets. So, okay, you mentioned about leaflets. I believe that just really means like, the top part and the bottom part, I guess.
Right.
And yeah, it shouldn't be just, in one of the whatever leaflets. And yeah, we have the trans one and the lateral one. Um, and and yeah, the lateral one should be easier, and that can, just be what is it? Something that happens fast or quickly.
So trans bilayer happens slower.
Mm-hmm. Yep. And I'm pretty sure it would need um enzymes like lipase and floppase.
I think I've barely studied that stuff.
Yeah. Yeah, you don't have to know too much about it. You just, we still were able to logic this out, but lipase would be like from the top down here to the one on the bottom. Floppase would be from the bottom up to the top. Um and then if both of these happen at once, that could be caused by something called a scramblase.
Oh my God. That I've never heard of.
Yeah, the scramblase, since it's like just flipping both, uh, it does not require energy. Um,
Um,
but a flip base and flop base, since they're just doing moving one thing, would require energy from the hydrolysis of ATP. So flip bases and flop bases, I think, would then be uh, not synthetases, but um, oh, they're just ATPases, I guess. But yeah. So, yep.
right. So next is this. I feel like it's super low yield, but I don't know. Like I have to know because there's so much there's so many of those pseudostratified, like
Uh-huh.
stratified, and there's like a lot. And there's also the type of cells they make up, you know?
Mhm.
Like nephrons are what type of appeal, and like I don't know, I feel like that stuff is so yield. Should I memorize it or
You said about the nephron part, or
yeah, I mean there's just like different epithelium makes up different cells, right?
oh, I thought you were talking about the nephron thing. But yeah, yeah. So yeah. So uh okay, so epithelial cell, obviously the one that's gonna be facing, the outside. Um and we can have, let's see what they're talking about. So they're talking about columnar, and those are like the long ones or the tall ones. What is there? There's like cuboidal, which are more cube-like shaped cells. Um, and what else do we have? We can also have the okay. Simple would be just one layer. Stratified means more than one layer. And now for pseudo stratified, it's going to appear kind of, let's see how I can put it. So, let me draw this. Okay, so let's see. Simple could be like, let's say like, okay, let's just say that these are these kind of cells, and then let's say these are, whoops, something like this, and something like this. So this is when it's flat like this. I didn't draw the nuclei, but for each of these are going to be a circle, of course, for the nuclei. So squamous, however you pronounce it, squamous is like kind of like flat cells that are like kind of flat. Um, this is cuboidal because they're kind of like squares or cubes. And then these like tall, long, rectangle ones are columnar or columns. Think of like a column, it like holds up like a pillar or something like that. Uh,
Yeah.
okay, and these are all simple, right? Now, stratified, if you think about the word, it means like layers and stuff. So you can have, let's say this, but just like more than one layer, and so this will be stratified squamish. Jeez. All right, whatever.
Don't worry, I got you. Yeah, yeah.
And then just imagine like now you have like two layers of the cuboidal thing, and then that'll just be stratified cuboidal, and then
Okay.
two layers of the columnar would be stratified columnar. Now pseudo stratified is going to look kind of like it's stratified, like but it would still be one layer. And so that would look like this. It would be like, so it's still one layer, but it's like this one can like kind of, touch like the inside or that part of the bottom part of this thing. Um, and then, this one on the next one here can touch, the top one here. So it's like still one layer, but it's also as if there's two layers, but it's really just one layer. Um, yeah. Here, I'm gonna put this in the chat.
Whoa, it's a monstrosity. Actually, okay. It's not that bad.
Yep. And so you see the simple ones are all just a single layer, and then the stratified
Yeah.
ones are all multiple layers, but the pseudo stratified, it's like one layer technically, but it's since you have like this one, right? It's almost like that is, it's facing this part, so it's like facing the inside, and then like, this one is facing the outside. So it's like it's as if there's more than one layer, but it's still one layer, so it's pseudo stratified.
Hmm. Also, the reason I mentioned nephron is because the simple cuboidal makes up the nephron. I
Uh,
was just saying, I was asking like
yep.
if it's worth to also memorize the location of all this stuff, because
Mm,
that's probably really low
yeah,
yield.
yeah. I haven't seen questions that are like as specific as this one. But I think just knowing, the difference of these could help. But yeah, the location probably not. Yeah.
All right, well, returning to the question,
Mm,
um, I would say A is correct,
okay.
right? Because they all touch the bottom
Let me see.
and you drew it.
Oh yeah, that bottom part is the basement layer, right? Uh,
Yeah,
seem to be touching that. Okay, that's a better explanation than what I was saying. Because if it's stratified, only the ones on the bottom layer will touch that bottom part, right? And only the ones on the top layer will touch the top part. But pseudo-stratified, all the cells touch the top and the bottom. So,
yeah. So wait, is that the correct answer?
uh, yeah, let me see the other choices. There are multiple. So B, if it's multiple layers, it will be stratified, right? And then C forms the discs. Oh, that's annoying because you would have to know what that thing is.
Exactly.
But yeah, I don't think it's gonna be in the vertebra.
It
But
says the intervertebral discs are formed of a connective tissue called fibrocartilage.
all right, so that's just a type of cartilage then, not like a, cell, like a, you know, epithelial cell. And then rapid exchange of large molecules. Hmm, I'm not sure what that could be, but yeah,
Mhm.
like we can at least rule out B and yeah, so A should be it. Yep.
Okay. It would just be a little difficult for me to do on an exam if I don't really know C and D that well, you know?
Yeah.
But
I'm with you on that. But yeah, I think it's something that's like on the low yield side because I've never really,
yeah,
yeah.
yeah. Do we just need to know Jacob, Monad, and operons? Just have that in our head as connected.
Um, yeah, I think so. Let's see. Yeah, I think that's what they showed, right? Jacob Monod.
Yeah. Okay. Yeah, next is I think this.
Function of the exonuclease activity of DNA polymerase. Excising a mismatched nucleotide following DNA rep. Okay, so well, Okay, what does DNA polymerase do? Well, just in general, without worrying about exonuclease activity.
Uh, Creates the complement strand of DNA.
Yep. So it basically just goes like, okay, here's a adenine, let me put it here. Here's a thymine, let me put it here. So that happens during replication, right?
Yeah,
So I don't think that it would be doing things after replication.
yeah,
So,
okay.
so I would think that A would not be the answer.
Hmm.
Um, okay. And well, B is during DNA replication. That's good. Let me see. C, replacing thymine dimers that form as a result of UV exposure. So, well, that's going to be after replication or, not during replication either, right?
Hmm.
And then D is also not during replication, it's during transcription. Um, what would do D?
I feel like it's right there, but uh,
So if this said replication, this would be right, right? But since it says transcription, that would be RNA polymerase, right?
okay, got it.
So yeah, I think it's going to be B simply because, well, the DNA polymerase does stuff during replication, and nothing else, no other choice says during replicate is about during replication. Yeah.
Hmm. Says that like the exonuclease activity is able to excise, remove, and correct DNA nucleotides during replication. Yeah.
Mhm.
Okay.
Okay. What is not a requirement for natural selection to occur, aka Hardy-Weinberg assumptions, right? Pretty much what it's asking.
Do I have to memorize those?
Yeah, I mean, you can kind of think about what would, let's see. Yeah, like I guess what would make it so that, you know, natural selection occurs, right?
And
So,
you want variation.
what's up?
You want variation.
Mm-hmm.
You want competition.
Mm-hmm.
That's how the adaptive traits get passed. Uh, yeah, between B and C.
Oh, you know what? This is okay, all right, wait, this is a little different. Because I know Hardy want so for natural selection. Because no emigration is a requirement for Hardy Weinberg, right?
Yeah,
So,
that. Yeah.
and I know C, like, genetic basis for physical traits is like a true thing, right? So
Hmm.
that would lead with B, but no emigration is a Hardy Weinberg thing, but it may not be a thing for natural selection to occur. Because natural selection is just essentially if you are fit enough to survive and reproduce, you're gonna, natural selection is gonna work out for you. Um, so emigration is fine because in Hardy-Weinberg it's not fine to have immigration or emigration because you need the population like to be the same, like not change for the alleles in the population. So yeah, this is actually not specifically Hardy-Weinberg here, just natural selection because yeah, because B should be your answer, and that is required as for Hardy-Weinberg, but not for natural selection. Yeah.
How about this? These words always get me confused.
Uh-huh. So 3 copies of the um, you know, chromosome 2 would be like so okay. The ploid part deals with the entire like sets. So like a dot so like we have so like for us 2n is well in general for anything, right? 2n would be diploid, n would be haploid. So for us haploid is 23 chromosomes and diploid would be 46 chromosomes, right?
Mhm.
Like in total. So triploid would just be 3n, right? But that's not really a thing. I think we did maybe a problem on triploid before, and you have an issue there because, you know, so for instance, for 2n, you can have, homologous chromosomes, having crossing over events with genes being transferred back and forth, right? And you can even have that for haploid where, it's just between the chromatids. But theoretically for 3, like you would have something like that, and it's not like an equal kind of no like equal crossing over events or anything like that, or like homologous, pairs or anything like that. So triploidy uh is less is gonna kind of make things weird. But ultimately, ploid just means that we're talking about overall sets of chromosomes, whereas somi is like somi, like to mean a single chromosome. Well, it'll describe a single chromosome. So if you had, so like trisomy 21, right? It's like the 21st chromosome, like would look like that, right? Whereas all the other chromosomes would just be, something like that or whatever. If it's if there's an extra copy of a single chromosome, like trisomy 21, right? That means there's 3 of the 21st chromosome. So yeah, so this one is talking about 3 copies of chromosome 2. So it's not talking about sets of chromosomes, but specific chromosomes. So it'll just be trisomy. It'll be trisomy 2 in this case. So somy refers to the specific chromosome. Ploidy refers to sets of chromosomes.
Okay. So what the answer here be? It would be trisomy.
Yep. 3 copies of chromosome 2. So it's just 3 copies of a single chromosome. So it won't be ploidy and it'll have to be somy. And if it's 3 copies, then trisomy. Yep.
Oh. If what would it ask if the answer be triploidy?
So like maybe they can say because it's the thing with the triploid thing is like I can't imagine it being like really feasible because of this weird thing with the crossing over. But So like my imagination would just be like if it just said something really like unnatural or something by saying like, okay, let's, cells from a non-viable pregnancy are tested and they found what is it, like 65 or something like that. I don't know, like 23 times 3, so like
Yeah.
69 chromosomes perhaps. Uh,
Hmm.
but let me see if there's any organisms that have probably some weird ones. Well, there's triploid syndrome, which really messes things up, messes people up who have it, and they don't seem to really be living or die uh, as infants and stuff. Um, but I wonder if okay, it seems like it's more present in plants and salmon. Uh,
Interesting.
wait, no. Salmon, apparently it only happens. So, okay, if you take fertilized salmon eggs and you give it a temperature shock or a pressure shock, it can induce it to start replicating a third chromosome set. Uh, so that's not something normal. And then for reptiles, I just asked ChatGPT. Reptiles, it's rare, but that it could happen. Some species of oysters, but it's very, very. It's not. It seems like it's happening like in a very, very like artificially induced situations or super, super rare kind of things. Because that's like the issue. Like you can't get like crossing over events here, right? Like here between the chromatids, you can have crossing over where something here can go here, something here can go there, right? You can do that for the homologous chromosomes here as well. But if it's three, it's a an odd amount. It seems like it's that's going to be weird. Because also, what's
Okay.
step afterwards is when these both of these chromosomes get pulled, right? The opposite ends, right?
Yeah.
And same thing with this, it would just be the chromatids, so it'll just be that for opposite ends. So, and that means in cytokinesis, you're getting two cells from one. So it makes sense to have an even number of sets of chromosomes because when it splits into two, one can have, they'll each have the same number of sets. But how can that happen for a triploid, right? It's an odd number of chromosomes. So how can that give you, an
Hmm.
even amount for both of the cells that it divides into? Can't, right?
Got it. Yeah, yeah. Okay. Next is this. It's just like a fact too. We gotta know.
Uh yeah, so the pentose phosphate pathway is an anabolic pathway because it's not breaking things down, but it's building, ribose sugars that can be used for like, you know, uh DNA nucleotides, RNA nucleotides, ATP, GTP, all that stuff. So, okay. And yeah, it uses, oh, and then it says oxidative phase, right? So if it's oxidizing something, if whatever answer that we're looking for is oxidizing something, we want, it means that it itself will be reduced, right?
Yeah.
You want to choose, something that is a reduced form of something. So, uh, yeah. So the ribose 5 phosphate, the zellulose 5 phosphate, those. um are products, but I'm pretty sure they would be during like um a non-oxidative phase. Um, so yeah, I don't think those would be it. Um, but yeah, it should be the NA.
Is that just like a fact we have to know?
Yeah, yeah, for the yeah, the pentose phosphate pathway is pretty short, like a couple of, steps, like a small amount of steps. So, and I have something in like the PDFs folder where I go over that. Um, but yeah, your end result's going to be the ribose 5 phosphate. But yeah, the way I was thinking about this was, oxidative phase means that we're oxidizing things, in the pentose phosphate pathway. So if it generates a product, that product must be something that got reduced. And that should be NADPH. And pentose phosphate pathway uses NADP and NADPH rather than just regular NAD. Yeah. Mhm.
So we just need to know that NADPH is during oxidative phase and ribose 5 ribose 5 phosphate is reductive phase.
Mhm.
Okay. Yeah, I didn't know that the initiation is the dendrites, right? Dendrites have the presynaptic neuron.
Um, let's see. So, um, sorry, okay, sorry. What are you saying? You're saying what about the dendrites?
The dendrites is where the action potentially gets initiated, right?
Um, let's see. I think no, I think that's where, so let's say, so the dendrites are these things, right? At the ends.
Yeah, yeah.
So that's going to be like, so I'm pretty sure the action potential starts at the axon hillock here because it travels down sorry, this is the soma, the cell body part. Um,
Yeah.
and I'm pretty sure that's where it happens. And then it passes that action potential down the axon, the long portion. And then all these like little like, finger like, projections there are the dendrites. And that's where the action potential can leave the presynaptic neuron, travel across this synaptic cleft here, and then enter into the axon, the soma of the next neuron. So the
It
synapse,
said
yep,
it got initiated at the axon hillock.
yep. Oh
And
wait,
so I was confused,
okay, okay,
like about because I thought it was Soma 2 originally.
yeah, yeah, okay, okay. JK, JK. Let's see. So, well, oh, axon hillock. I was comparing that to the dendrites, which are all the way at the end. But the axon hillock is where I drew, or this part, like. the part where the long portion kind of starts and the cell body part kind of ends. And,
Okay.
um, so I actually think that okay, no, I'm gonna so I was so the axon so sorry, the dendrites are actually the finger-like kind of projections of the soma. So these are the dendrites. This is the soma. This is the axon, the long thing. And then right here, where the soma and axon meet, is the axon hillock. And then all the way at the end of the axon are axon ter terminals. Yeah. So yes, axon hillock is where it starts. So okay, cell body of the neuron is called the soma. True. Synapse is the site of the initiation of the action potentials. That's like, you know, this gap between like, the axon terminal of the presynaptic neuron and then like the dendrites of the postsynaptic neuron. So that's not true. It's not synapse. It's axon hillock. And then C, neurons internally transport neurotransmitters along micro tubules. So like the neurotransmitters could be like acetylcholine, things like that. And yep, that can be transported within the cell. And typically what transports things in a cell are microtubules, right? So that should be true. And then neurons have synaptic knobs filled with neurotransmitters. Yeah, they're able to internally transport neurotransmitters. They should have like, well, when I say knobs, I
I never heard of that term. Do they mean like the vesicles?
think so. I think I'm just thinking, I guess, of like kind of like this part over here. Um, but let me see, synaptic knob. Yep, it's basically like if I zoomed in on just one of these axon terminals, it would look like that, right? And then let's say this is like the next thing. So this like bulb it also looks like a knob, like a doorknob, right?
Oh, okay. I thought nodes were like the actual vesicles
I
with
mean,
the neurotransmitter.
oh yeah, yeah, those would yeah,
Okay,
yeah. Mhm.
got it. Okay. So it's just a fact that it gets initiated at the axon hillock.
Mhm.
Interesting.
Mhm.
But doesn't the soma receive the signal?
Mhm.
Like,
Yep.
but I guess it doesn't initiate it.
Mhm.
Okay. So in terms of like body systems,
Mhm.
I really lack in the in like MSK, like musculoskeletal
Mhm.
system.
Got it.
So yeah.
Yeah, so the sarcomere, um, what can you tell me about a sarcomere?
Uh exists in muscles. So it has a there's like T tubules that transmit electrical impulses, and then there's the actin-myosin. Um, there's it's just all mixed up right now in my head. I mean, like I know some of the facts, like the M line, the Z line, the T line, whatever, all that. But that's like I just don't know how it comes together, honestly. Um, the only thing I know, the main thing is the whole pathway of like CA getting released from the sarcoplasmic reticulum when acetylcholine binds, and then CA binds. It opens up the whatever you know what I'm talking about, like tropomyosin, troponin, and actin-myosin.
Hmm.
That I know, but sarcomeres and how they like stretch or whatever, shorten, I don't know well.
Hmm. Got it. So I kind of drew something here uh, that's supposed to resemble a sarcomere, and we can have our well, myosin and actin, right? So
Hmm.
the myosin and like this is this is like the actin,
Yeah,
and you'll do that cross bridge cycle, which will attach these two, and then some type of power stroke that kind of moves it so that
yeah.
the actin it can move to the left and the myosin can move to the right, or like more so like stretch to the right. And so as this happens, it's going to look more like that, where the muscle contracts and the sarcomere gets shorter during yep.
But why is that? Like
Mhm,
when it when it the actin pulls the myosin or whatever,
mhm.
or no, yeah, I thought myosin attaches to actin.
Yep. So the actin here, the myosin here, and I just drew this kind of thing over here to show that it connects. And, and if this moves, whoops, or bends, that way, it will cause the actin over, I'm gonna use blue. It's gonna cause the actin here to go this way and the myosin here to go that way. So you get more overlap of the actin over the myosin, right? But the overall sarcomere, which is this entire thing, will shorten
Hmm.
the muscle contraction. Okay. So now the thick filament is the myosin, the thin filament is the actin, and we see that they're both present in a sarcomere. So we can't say that they only contain thick or only contain thin. So those are out. Now the overlap happens with the myosin and actin overlapping. But the sarcomere is this entire unit, and that entire unit isn't overlapping, right?
Hmm.
So C is wrong because it says sarcomeres overlap, and that's not the thing that overlaps. It should be the myosin and actin that overlap. So C is out. And yes, when the sarcomeres shorten, which is what happens during muscle contraction, the entire muscle shortens, and that's why we call it contraction, right? Um, when you say the opposite, which is relaxing, that just means that it gets longer or goes back to its default length. Uh, but yeah, that's the idea.
But do we need to know anything about the A band and the H band and all that crazy stuff?
Let's see. So the okay, so the ends will be Z. Whoops. Oh yeah, there we go. So you can see here that the Z line is what demarcates one sarcomere from another sarcomere, right?
Hmm.
In the drawing that I made like this, oops, like this and this are the Z lines.
Okay.
You can even see like if you look at like how there's a smooth muscle.
Okay, yeah.
Um, but uh, other muscles, striated. The striations are caused by the Z lines here. And so they come up as these vertical lines in like some, if you look at it under like an electron microscope or something like that. Um, and that's what causes the striations in muscle. And yes, those are the Z lines. The M line will just be that one right in the middle. The A band, just think of the A band as being the entire length of the myosin, which is this over here. The myosin, which is the thick filament. So the length of that myosin from this end to that end is the A band. Okay. Um, and then the H band is from the end of this actin and start of that actin. So it's essentially the gap between the actins because this blue stuff, these are all actin, right?
Yeah.
So yeah, so basically the H band just represents uh part of the gap between the actin stuff, the actin ones. And then the I band is from the end of the myosin to like over here should be the next myosin. It'll be like red to that one. So that includes the Z disc as well as, so this coiled thing here is called titin, which is essentially the thing that when you're stretching, like that's kind of what you're stretching. And this is I think the longest or largest like protein, or at the very least, the name is the largest name for like anything. But yeah, I think that can kind of help you out. So if you think of the Z line as just what separates, one sarcomere from another. So these are the Z lines, and they're visible under like a microscope. The gap between the actins here is your H band. And when the muscle contracts or shortens, right, you eliminate that H band, or at the very least, you shorten it. That's what we have here. Because during the contraction, right, this, see how it's like kind of bent at this angle? It
Right.
it gets, so like this is, oops. So like this is actin, this is myosin, and it binds like this. And then what happens is this thing gets like goes from like an angle of like 45 degrees or something like that, or just so let's just say this angle to that angle. So it has this motion. And so as it has that motion, since it's attached to the actin, it pulls the actin forward or to the left and the myosin to the right. So then it looks like this, where you have the actin now moving to the left and the myosin moving to the right. So as a muscle contracts, the H band shortens, um, and then the A band is just like the length of that spans this myosin filament. And since it's just the myosin filament, that doesn't change because the myosin is not, uh, really being moved here. Um,
But like how high yield is this stuff? Is it have you seen it on AMC things? Like
so
specifically H band
yeah,
and I band.
yeah. So I haven't seen any question that asked you specifically like to differentiate the A band from the Z band line to the I band to the H band. Don't yeah. I
Mhm.
see any questions about that. But what you're expected to know is what a sarcomere looks like. Like if you're able to draw it like this, like that, and you understand that the myosin is like this thing, and then like the actin is that stuff, and then you understand the cross bridge cycle of how with the power stroke and stuff, like how the myosin head can attach to the actin, and then it can get cocked or go from like 90 degrees to 45 degrees and then causes the muscle to shorten, like that's what you're expected to know. But yeah, you're not expected to know like specifically the difference between an H band and an I band and stuff.
Yeah. Okay. This is this is all kind of basically burnt into my head at this point, I think.
Hmm.
So yeah, I think this I should have down.
Yeah, so you see this is better, more accurate in terms of compared to that other one. We start when we first bind, when the myosin head first binds to the actin, right? It's a 90-degree angle here. Okay. And then when the, inorganic phosphate is released, it goes into this 45-degree angle, right? And so if something goes from this 90-degree angle to this 45-degree angle, that means that this moves that way, right?
Yeah.
And so now if it's attached to something, when it moves this way, it's going to take this thing and move it forward, right?
Mhm.
So here, cross bridge formation, and then this is called the power stroke. So you see how the angle of this is smaller because it goes from 90 degrees where it attaches to 40. And so when it attaches, when it goes bends from 90 degrees to 40, you see my left arm here, it's supposed to be the actin, and it goes like that. It pushes the actin to the left.
Mhm.
So that's why you get the actin stuff get closer together. So, and that's contraction. So, uh, yeah.
Um,
Mhm.
let's do this real quick.
Mhm.
I I remember this question. Yeah, we actually might have done it before, but just while since we're on the topic, I
Yep.
want to go over it.
Mhm. Skeletal muscle sarcomere is shown above. The ability of a resting sarcomere to contract is dependent on the existence of an overlap between actin and myosin filaments and the presence of a gap between the ends of the actin filaments and the M line. Given this, which of the following diagrams best represents a resting sarcomere with the potential to generate the greatest amount of contractile force? Okay, now I want to see what they mean by the resting sarcomere. Okay. Resting sarcomere, when it contracts, is dependent on the overlap between the actin and myosin filaments. So, greatest contractile force, I would imagine the greatest amount of overlap. But it does say that there still must be a gap between, right? So, I'm unsure about how far because these overlap such that there's no more gap between.
What does it mean by gap?
My guess is the H whatever or what I think it was called that H zone um is that gap, right? In that other picture that you have in that the bottom thing was showing during contraction that the H gap was still present, it was just narrowed. So
The thing is, it says resting sarcomere. Isn't? isn't B already contracting a bit and D also?
Yeah, I was like wondering like what they mean by the resting thing. But which of the following best represents a resting sarcomere with potential to generate the greatest contractile force? So when it contracts, um, like I guess, how much the overlap in the presence of a gap between that and the M line. So let's see. Oh, well, okay, so I don't think it'll be B or D because there's no gap there anymore and it's already like the actin stuff is already overlapping. And between A and C, like I guess this one has a larger gap, so there's more potential for like, this thing to move here and this to move here, whereas this is just a smaller gap. And if they're saying that the um ability of a resting sarcomere to contract is dependent on the existence of the overlap between actin and myosin and the presence of a gap. So let's see. There's no overlap between actin and myosin in C.
That's the thing.
Mhm.
How's it gonna like move them? How's it gonna contract?
Um, yeah,
You know?
that's a uh good
I'm
question.
just playing devil's advocate. I'm trying to like reason this through.
I was initially gonna think like A, but I was also looking at C because there's more of that
Yeah.
overlap. I mean, sorry, more of the gap. Um, so so like C has much more potential for the contractile force, and I think that this maybe could constitute because we have like these angled, kind of in such a way that
Yeah.
maybe it, can bind there and then pull it that way. So I guess it's C. Damn it.
Okay.
All right, I guess then, that is not really like overlapped. But let's see. Actin-myosin overlap and C, I guess doesn't have that overlap, right? So yeah. So like
C's. Mm, yeah.
yeah. So yes. I think that C, when you described it as not having the overlap, I was thinking, okay, that means that it can't be it because the myosin head won't be able to bind to the actin. But I just kind of like just kind of made up like a connection here because I thought that I just really looked at it like in a zoomed in sense.
Apparently it's excessively stretched.
Hmm.
And it doesn't have that overlap. Yeah.
Yeah, yeah. Like not having the overlap would have been the giveaway for it being A, but I
Hmm.
like looked at
No,
it,
I get what you're saying. I get what you're saying. That little yeah.
yeah, and like
Okay.
right? Like if you took a ruler and you just went down like this, I was like, okay,
No, I get.
maybe it's like
Okay.
last part of it or whatever, right? But
Okay.
yeah, yeah. Otherwise though, for that question, it gave us
Yeah.
all information though. It did tell us that, and the most important information that it gave us was that there must be that gap, like even during contraction. So that's why we were able to eliminate B and D.
Got it.
Yeah.
Okay. This is this.
Okay. All right. So yep. What do you like? What's your approach?
So when Blot detects DNA, it can detect a specific DNA sequence.
Mhm.
Um, yeah, four unique fragments. Um, I mean, like I did 4 factorial and I got 24, but like I don't know why it's that. I also have never used factorial in an MCAT question except this. I don't know.
So
I
I guess
just
that could be like the oh wait, okay, let me see. Southern blot of the digested DNA sequence and detected four unique fragments. How many DNA sequences could the observed fragments have been derived? Okay. Okay, I think that maybe. All right, so any type of blot, right? You're going to, so to digest the DNA sequence, we're going to have to cut it up into the, let's say A, B, C, D. One, two, three. You cut it three times to get four fragments. Um, and then how many possible sequences can those fragments come from? Um, how many possible? So, okay, if each of these fragments are unique and different, um, how many possible DNA sequences been derived? I'm thinking like four, maybe, like just because they're talking about DNA sequences and
answer is D. Answer is D.
it's D.
Yeah, it's 24.
Okay, so I guess it is the factorial thing.
It is. That's the way you do it, but I just like don't know why.
Oh, okay. I could answer that because what I was originally thinking was like that 24 isn't the answer. So I was trying
Yeah,
to,
no worries. Of course. I shouldn't mention it. It's my fault.
but
But yeah, it is the answer.
so let's say we have four slots, right? And um, you know, A, T, G, C, right? So let's say that for this first slot here, it can be any of these four nucleotides. So that can be four possible things, right? Um, and so if these are the four unique fragments, right? Meaning one can be just A, one can just be T, one can just be G, one can just be C. So yeah, the first one, because from the original before it was cut into the four different unique fragments. So the first one could be any of these. So that'll be four possibilities. Then, one of these would be used. Like, let's say it's A. So that's out. So the next one can be T, G, or C, three options, right? And let's just say it's T, right? So that's out. Then the third one can only be G or C. So there's two possibilities. Let's say that it's G. So that's out. Last one can only be C. Oops. Can only be C. And that's just one possibility. So that's why it's 4 factorial. Um, so it's just a yeah. So that's why it's 4 factorial.
Okay.
Yeah.
Next is this.
Okay. Real time PCR can be used for all of the following purposes except for um well, okay, what's another type of PCR that maybe comes to mind?
I don't know. There's PCR, qPCR, RT-PCR.
PCR?
What?
What's qPCR?
I don't really know.
Um, what could it be if it starts with a Q?
Quantifying.
Yeah, yeah. So I think that, well, wait. No, no, no. Okay, no, no, no. Got it, got it, got it. So so PCR is about figuring out stuff about nucleotide sequences, right?
Okay.
Like particularly for RNA, right?
Hmm.
Um, so so it can help us detect a specific nucleotide sequence. It can help us quantify gene expression because uh, DNA transcribed to RNA is what starts that gene expression. Um, and then identifying the presence of a mutation you can also do. But protein levels is going to require the RNA to be translated, by ribosomes to turn into proteins. So real-time PCR, which is, helps us figure out or look into nuclear like RT sorry, like RNA, DNA and stuff like that. But it can't tell you anything about protein levels.
PCR, you just like make a lot of copies of DNA.
Yep, you amplify yeah. It can be DNA, RNA would be like uh also that. I'm thinking most of the time I think it's RNA to be honest, but yeah, we're trying to look at DNA, RNA, uh, you know. I'm pretty sure it's really just RNA. Let me see. Yeah, it's specifically RNA.
Uh,
Um, yep. So so yeah, yeah. Protein levels we cannot figure out from RNA because it has to be translated into to turn into protein.
mhm. Okay. There's this other question I want to do.
Mhm.
And yeah, this should. Um, our first and probably last psychology question.
Mhm.
I was really confused with this, and I think like very little amount got this right too. So this is in regards to primary appraisal.
So I'm thinking of that in like one of those cannon or
Yeah, it's yeah, yeah.
so okay, let's see. So
This is Lazarus, I think.
what was that?
Uh, actually, I don't know. Does it have to do with Lazarus theory of motion? Appraisal? Appraisal theory?
Oh yeah, I didn't even know what that was. I think he showed me something like that because I only know like the three, like the James Lang, Simone something, and
You got
Cannon.
a 132 and you
Cannon.
didn't know what this was?
Yeah, I didn't know that there was like a third one because I've never seen,
Wow.
it was
Okay.
the MCAT books that I've seen. I've only
Hmm.
even now that I look it up. I only see I see James Lang, Cannon Bard, and Shachter Singer.
Right.
But you're saying that there's a third one, right?
Yes. Lazarus serial motion,
Got it.
event, you label the event, which is appraisal, and then you have emotion and physiological response at the same time.
Okay.
So, anyways,
Mhm.
asks which is primary appraisal. Yeah, I just had no idea, I mean, how to go about this.
Got it. Okay. So, like appraisal, I guess, would be something like, you know, can I perhaps okay, let me see. Checking your calendar to see whether you have any appointments with hostile clients, asking yourself whether you feel calm enough to perform successfully in a debate, looking out the window and deciding whether you'll be able to drive safely on a dangerously icy road, whether you're alert enough. Um, so the appraisal, let's see. Hmm.
So apparently B, C, and D are all secondary appraisal,
Hmm.
which is when you assess your ability to cope with the situation.
Okay. So primary, yeah. Okay. So in terms of pattern recognition, I guess, right? Um, Asking yourself whether you feel calm, deciding whether you'll be able to blah, and then whether you're able to do something there as well. Whereas A is the only one that isn't asking, am I capable of, handling this? It's just straight up, is there going to be any threats or something like that? So
Okay.
yeah, that's cool. Good to know that. Makes sense. Um,
Hmm. Yeah.
so uh so yeah, so okay, primary appraisal, is this a threat? Um, Secondary appraisal, am I, you know, I don't want to say qualified, but am I ready to face this threat?
Yeah. Yeah.
Yeah.
Okay. Sounds good.
Mhm.
Um, yeah, we end right on the dot when we started. Okay.
Mhm.
So we have a session tomorrow. I'm gearing up to take the first blueprint full length. Well, my first full length, I mean,
Wait.
on Saturday.
Oh, okay.
Like in a while. Been 2 months since I took a full length.
Wait. Um, did you mm, not are you not the section bank?
What about it?
Yeah, when were you planning on?
Well, I don't have a date when I'm planning on finishing it. Um,
'Cause
but I
yep.
I think at this point though, I really want to take a full length
Mhm.
just to well, it's I've been studying technically for a month and a half now. Technically, I really ramped things up the last like probably 2, 3 weeks.
Mhm.
But like, the thing is, I really want to see where I'm at because if anything, it'll be a push to study even more seriously. Because if I keep delaying it, it's not gonna be good. And the other issue is like there's only an X amount of Saturdays where I could take the full lengths at this point. So yeah.
Got it. Yeah, I mean, because yeah, either you take the full length on the 12th, this Saturday, and then next Saturday you're not taking one, and then the 26th you're taking one. So so I would either think to not take it this Saturday, complete the section bank, and then take it next Saturday, and then the Saturday after that where you don't get a break in between. But it's up.
That's the thing. Next Saturday, like I can't. I'm doing this volunteering thing. And then
All right, so you
um
could take the third party, sorry, yeah, you could take your next, your blueprint full length, and then we can just decide some other time to do the section bank, because we're gonna have a ton of time in between tests, right? And
yes.
so, okay. So let's see. So you're gonna do the blueprint one, and for cars, are you gonna like switch over to something else? Like,
Yeah. I'll do like 9 Jack Weston passages.
okay. And forgot for the AMC cars, QPAC 1, QPAC 2, diagnostic tool. How far are you with those? Have you
Been doing those like I just follow the Jack Weston cars course
oh,
every week. I've been doing a decent amount of those already. So
got it. So okay. So you'll use Jack Weston.
yeah.
If you want, you can use the AMC ones as well. Okay. So yeah, okay. And then yeah, tomorrow, yeah, what do you want to do tomorrow?
We'll meet tomorrow and yeah. What I want to do tomorrow is just go over some other topics that like I've been struggling with.
Mhm.
So you wanted me to explain to you electrochem.
Oh yeah.
Maybe yeah, maybe explain to you optics just so I mean I have to make sure I retain that from like 2, 3 weeks ago.
Uh-huh.
I'm gonna review it
Yeah.
tomorrow before the session. And I also want to go over buoyancy, whatever. And
Mhm.
then acids, bases if we have time. But
Yeah.
yeah, those.
Awesome. Okay then. So yeah, so okay. Just like 2 hours, so yeah, 150. And
Okay.
see you tomorrow at 5 p.m. Eastern.
Yeah. Sounds good.
Awesome. See you tomorrow.